Hello everyone, and welcome to our online lecture series for Bio 1. Today, we begin our section on organismal biology. In our last lecture, we talked about how a cell goes through its cycle in order to perform mitosis and grow. And it's quite fascinating how all these different organisms, the absolutely giant and glorious elephant, the cute, cuddly, lovable, obedient dog, the ever handsome human, and the quite complicated and regenerating axolotl all begin from a single cell, that diploid zygote, that will then go through one round of mitosis, and again, and again, and again, up until they reach their respective sizes. In the process of performing mitosis, we go from that single zygote that is unspecialized to many different cells that correspond to many different tissues. These tissues in animals, of which there are 4 tissue types, are utilized in different patterns to create the amazing amount of variation in terms of body plan that we see in our different animals. The variation in body plan correlates with tissue diversity, meaning that animals that are very simple in their body plan have fewer different tissue layers and different tissue types. The different tissue layers are as follows. They correspond to the development somewhere along the lines of when you were a tiny zygote. So after something in between the single cell and then the multicellular entire organism, when you were still a small ball but had many cells in that ball. In the development of that ball, the most complex organisms can develop 3 different tissue layers. The outside being the ectoderm, the middle component where organs develop, the mesoderm, and then the internal lining of your digestive system, the endoderm. And these are our 3 tissue layers. These layers correspond with different axes of symmetry. The first and simplest type of symmetry that we see in animals, and yes, that is an animal, Spongilla lacustris, or the freshwater sponge. It gives us an example of asymmetry. It has 1 tissue layer. So none of these different components need apply. It's 1 different tissue layer with a general blueprint of 1 tissue and cell type. It's just the same repeated thing over and over and over again. You can't cut it in any axis and get a similar pattern on either side. It does not repeat. It has no pattern to its growth. So sponges are our most simple animals. They come from the kingdom Porifera, or they are in the class Porifera. Also, I didn't mean class or kingdom. I really meant phylum. My apologies, y'all. Is the phylum Porifera. Now, once we start getting more time in between of organisms like sponges existing, a certain subset of them served as the descendants or served as the ancestors of our next group, a group that does have a pattern, a group that shows us radial symmetry. These organisms with radial symmetry are in the phylum Cnidaria, more commonly known to us as the jellyfish. Now, cnidarians, with their radial symmetry, have multiple axes of symmetry. You can cut them vertically, you can cut them horizontally, diagonally. In each of these axes, from the top-down orientation, they will be symmetrical. Hence, they have radial symmetry. In radially symmetrical organisms, you get several or two of these three tissue layers. So I'll copy this back down here. You do create the ectoderm and the endoderm, but the mesoderm, where organs would develop, does not exist. So radially symmetrical organisms, like jellyfish, only have the two layers of the ectoderm and the endoderm. No place for internal organs to develop. Our last group has all three and is bilaterally symmetrical. These organisms, like our very handsome human, illustrated by my fantastic drawing skills, I'm sure, can be cut down a single axis to show their symmetry. From the head to the spine. The formation of the head itself is something unique to bilaterally symmetrical organisms. They went through the process of cephalization, or the formation of a head at the anterior end of the vertebral column. That leads us to our words of anatomical location. In order to illustrate this point, I will draw a very majestic horse. I know, right? The side with its head is known as its anterior. The side at its tail is known as its posterior. Hence, why your first grade teacher might have told you to sit down on your posterior. Many animals with this vertebral column have what is called a post-anal tail, meaning that there is some sort of appendage after the anus used as a stabilizing feature. Everything from horses to our majestic canine friends to elephants and the ancestral condition in our amphibian and fish relatives have that post-anal tail. So if the top of the spine is the anterior and the bottom of the spine is the posterior, the above spine is known as the dorsal side, and the in front of spine is known as the ventral side. So it's important for you to know dorsal above, ventral in front of, posterior at the back, anterior at the front, and that the post-anal tail helps orient all these things. All organisms to which these terms apply have those three tissue layers that we see in bilaterally symmetrical organisms with cephalization, or the formation of a head. Now, all animal sizes, because I will admit, as much as I'd love for my dog, and yep, that's little Odie, as much as I'd love for him to be about the size of an elephant, He is not. Oftentimes, animal size is limited by energetics, or the amount of energy needed to maintain that organism. Not only is size limited by, jot this down. Not only the energetics or nutrients available to them, but all these organisms have variation in their oxygen delivery and, in general, nutrient delivery. And these principles keep animals limited in their size. For example, in a time in Earth's history, oxygen levels were much, much higher. This was during the Carboniferous. During this time, when oxygen levels were much higher, insects needed far less efficient oxygen delivery systems because the air was doing it for them. What that means is that insects grew in size to the point where dragonflies could have 4-foot wingspans. And you could get a centipede that might be 20 feet long. So be quite thankful that oxygen delivery does limit the size of certain organisms, like insects. And know that that is also a governing feature in size limitations to dogs, which can get as big as mastiffs, and elephants, which are quite gargantuan in their size. There are a couple other features about the energetics of animals that I want to get into before diving into those different tissues. A couple of vocab terms. By this point in the class, we've thrown this word around a fair bit: cold-blooded and warm-blooded, or the scientific way, the cold-blooded ectotherm, which is defined as an organism that gets its heat from the environment, and the warm-blooded endotherm, which gets its heat from inside, like you and I get those warm and fuzzy feels that happen to keep you alive. Now, in terms of energetics, an organism that needs to generate its own heat needs to have a much higher metabolism. So endotherms need more food, are less efficient with their food, and have a higher rate of cellular respiration than our ectotherms. In fact, you as a human, without moving at all, just by breathing, just by existing, and I'm sure we've had those days where the couch is awfully comfortable and we want to move nowhere, you still have a standard metabolic rate, or the amount of calories that you burn per day, of 1,300, that's supposed to be a 3, of between 1,300 to 1,800 calories per day. The bigger the animal, the bigger the standard metabolic rate. So elephants obviously need to eat more than we do in order to maintain their massive size. Now, some animals go into these fantastic states that allow them to lower their standard metabolic rate for periods of time. That alteration of metabolic rate is known collectively as torpor, where organisms can downregulate their metabolism to survive in tough times. For example, during the winter, some of us humans might feel like it, but we can't actually do this. But our bear cousins are able to hibernate. or slow down all of their metabolic processes to require less food. Now, this keeps them alive through the winter because there's not as many calories around. So they just take a really long extended nap where their heart rate is lower, their brain function is lower, and their entire metabolism does not need to go out and get food because they've packed it away to store it during this hibernative state. Alternatively, you can also get organisms that lower their metabolism when water is very low. We call this estivation. Hibernation and estivation. And you can imagine that there are organisms that live in the desert that need to be able to estivate when there's no water, and then might rise when they feel some rain a-coming. Now, the whole reason that energetics matters and our ability to keep track of our calories and our endothermy matters is because it all serves to get us closer to our state of homeostasis. And homeostasis is our ability to keep constant our internal conditions. Now, we need to keep constant our internal state at an equilibrium, not because it's comfy, but because it's necessary for protein function. We are fine-tuned. Every protein in our body operates with some given lag time and some performance time. And everything is queued up in such a way so that at our internal body temperature, every firing is coordinated. Meaning every launch of every neuron, every launch of the muscle is tuned to perform perfectly at our internal body temperature of around 98.6 degrees Fahrenheit. Though do know that that internal body temperature does fluctuate. Our proteins have evolved to perform optimally at this body temperature. Similarly, my dog's body temperature is optimized so that way his proteins perform optimally at his body temperature, which is actually somewhere between 101 and 102 degrees Fahrenheit, so a little bit hotter than humans. So each animal that's endothermic has some optimum body temperature that's specific to allow its proteins to fire at their best rate. Now, the way in which we maintain our body temperature is through a negative feedback loop, one of the two major ways in which we maintain homeostasis overall. A negative feedback loop will be familiar to most of us because in a negative feedback loop, your body will respond when it gets too hot by trying to cool down, or when it gets too cold by trying to heat up. You're always looking for that negative stimulus, that one that tells you that you're off your optimum. In this same way, blood sugar is regulated in a negative feedback loop. Food is consumed and digested, causing blood glucose to rise. I certainly would eat that pizza, but I have yet to meet a pizza that I would not eat. That pizza looks quite delicious. When food is consumed, the higher blood glucose levels in the blood cause the pancreas to make insulin. So as your blood sugar goes up, your pancreas corresponds by producing insulin to bring it down. The glucose is transported into red blood cells and into the liver. Glucose levels drop. In response to lower concentration of glucose, the pancreas will then stop secreting insulin up until you throw another pizza at it and make it do work. Another possible way of regulating homeostasis is through a positive feedback loop. Now, whereas in a negative feedback, gotta spell things correctly. Whereas in a negative feedback loop, you always wait for some negative response to send you towards the opposite. In a positive feedback loop, when something begins happening, more and more of it continue to happen. So it's kind of a snowball effect. We see the snowball effect occurring in clotting factors, like when you need to make a scab, as well as contractions in childbirth. When contractions start, they keep feeding contractions up until a certain individual, or individuals in the case of twins or triplets, are evacuated from the scene. Now, in terms of endothermic organism temperature control, there are a couple of different ways in which animals that are endothermic or warm-blooded have a way of controlling their internal warm-blooded body temperature. The first is insulation. Now, it might seem kind of strange to us because we have large stores of fat right underneath our skin, but the purpose of that fat is A, to provide us energy somewhere down the line when we need it, but also at the same time to help us maintain our internal body temperature. In organisms that are ectothermic, like, say, frogs, fat is not stored underneath the skin. It is instead stored in very strange-looking organs that look kind of like spaghetti throughout the body that store fat for the frog, but do not provide it an insulating factor. Again, major difference between warm and cold-blooded. Now, my dog also, in addition to his layer of fat, he's got a layer of fur, hair, made of the protein keratin. Does a great job insulating things. This allows Odie to go outside without a jacket during the polar vortex. Oh, he did not like the polar vortex too much. Additionally, we also have some neural controls, meaning that you can do things, or your brain can do things to help control your temperature. Most notably, vasodilation, meaning expanding the amount of blood flowing through your veins to either help heat you up or cool you down depending on what's being vasodilated. This is because where your blood is flowing changes your heat exchange. And heat exchange is a very important topic for our ectothermic organisms. Now, my dog happens to do this as well, but a lizard you can often spot sunning on a rock, absorbing the radiation of the sun, warming up. One way in which you and I cool down is through evaporation of water on our skin. You can additionally lay down on a hot surface, this heat exchange being called conduction, or get yourself a nice cooling breeze and cool down via convection. Now, ectothermic organisms only have these methods of heat exchange, whereas warm-blooded organisms, or endotherms, can do a lot more temperature control and regulation of their metabolism in order to drive heat up or down. So that gives us the background that we need in order to understand general body plans with symmetry, location, and the basic principles of energetics and homeostasis, so that way we understand why our different organ systems are doing what they do in order to keep our internal conditions ideal and very different from the external conditions. So let's end this lecture by going over those 4 tissue types. So where we originally began with those 3 tissue layers, we'll now explore those 4 tissue types that are found in everything from elephants to very cute puppies to artistically drawn humans to axolotls, these salamander cousins. The first tissue type is epithelial tissue, or skin tissue. This is the stuff that we use to line the outside of our bodies as well as the outside of our organs. Epithelial tissues can be defined as either simple, meaning that they are 1 layer thick, or stratified, where they are multiple layers thick. These epithelial tissue types include squamous cells, cuboidal cells, columnar cells, and transitional cells. Squamous cells are perhaps the ones that you recognize the most because they make up your skin and mouth. And then you have squamous cells within your lungs and blood vessels or capillaries. Cuboidal epithelial cells are those cube-shaped cells, and we'll be looking at cube-shaped cells later in this class, that make up our glands. They oftentimes are secreting hormones to the rest of our body. Columnar cells are perhaps my favorite because they make up our digestive tract. And each of these cell types is named for their shape. Squamous, like squished. Cuboidal, like cube. And columnar, like a tall column. These different tissue types, small emphasis on transitional, but they're really only found in the urinary bladder, serve as the lining for our bodies. So epithelial tissues are very important, and we will be looking at them under the microscope. Now next up, we have a quite diverse group of tissue type, that being the connective tissues. Now, as a tissue type, we have large variation in the different cells and fibers that make up connective tissue, because connective tissue is different than epithelial tissue because it is made of a matrix of living, aka cells, so living things, and non-living protein. We oftentimes call that non-living matrix the ground tissue in our connective. Now, the matrix itself, the fact that we have this web of living cells and non-living proteins in a mesh, is what gives connective tissue its density. And that is the defining factor about connective tissues. They're quite dense. The fibers that define connective tissue are of 3 different types. You can have collagen fiber, that provides strength. You can have elastic fibers that provide flexibility. Collagen's actually the protein. The elastic fibers are made of elastin. Very creative with this naming process, I know. And reticular fiber, or repeated fiber. And this is what we use to connect organs. Now, these fibers are made by a fibroblast. This blast suffix is something that we'll see a lot, but generally the fibroblast makes the fibers of our connective tissues. It's interesting because it is actually motile, meaning it can move around the body. So fibroblasts can move around and lay down track or lay down fibers in order to build our bones and other connective tissues. They also can split mitotically. And they are responsible for making all of our different types of cells in connective tissue. Now, the types of tissues within our connective tissue include the loose connective tissue, of which we see collagen, elastic, and reticular being some of the major drivers. And then our fibrous connective tissue. This is the stuff that we'll see in our tendons and ligaments. Now, tendons are notable because they join two different structures together. Tendons are muscle to bone, and ligaments are bone to bone. We also, in this class, will be looking at cartilage under a microscope and the chondrocytes corresponding, as well as some compact bone with its osteoblasts, the cells that make it up. But then two connective tissues that are oftentimes overlooked are adipose, or fat, and red blood cells. After all, where are red blood cells made? In bone marrow. They're stem cells that make red blood cells derive from the inside of our bones. They are connective after all. So here's a quick look at a cartoon of our compact bone that we'll be looking at later under the microscope. So you see that there are actually blood vessels in each osteon of the bone, the functional unit of the bone itself. We know that the bone heals, hence why it has blood vessels, living tissue. We also can see the lacunae, so this is what it'll look like under the microscope, each of the individual pockets within the osteon, and a more cartoon version of what's happening even in further detail. So connective tissue is quite diverse, but really defined by these three different types of fibers and its ability to be both dense and connective. Now next up, we've got our different types of muscles. Now there are three different muscle types: smooth, skeletal, and cardiac. They are defined both by whether they're striped, how many nuclei they have, and how we control them. Smooth muscle has no striations, hence its name of being smooth. It has a singular nucleus frequently and is involuntarily controlled, because smooth muscles lines things like your digestive tract. Now, fortunately, I hope, you've never felt your body moving excrement through your insides. That's because all of that movement is controlled with smooth muscles. In contrast to the involuntary, non-striated smooth muscle, skeletal muscle is striated, has many nuclei, oftentimes at the edges. So these cells can be quite huge, and they are voluntary, meaning that you control them. So as much as you want to say, perhaps to your parents when you were younger, I lost control of my fist when it punched my little brother in the gut, that never happened. You have full control over your skeletal muscles at all times. Yes, there's always a circumstance in which you lose them, but in most cases. Didn't work on my parents. Mom was a biologist. Our cardiac muscles are found in our heart, and they are striated and thankfully involuntary, and frequently will have only a single nucleus. So you have these cartoons of what these cells look like with excellent staining. These intercalated discs provide the striations that we see in cardiac muscles. And we'll talk later about the protein structures that give these skeletal muscles their ribbed features. The sarcomeres that allow for the push and pull that we see in our skeletal muscles and in our muscular tissue type. Now our last tissue type is that belonging to neurons or nervous tissue. We won't spend a lot of time talking about it today because they are incredibly complex, and we'll have 2 full lectures devoted to the nervous system and specifically nerve cells coming up. But you should know that the functional cell type of your nervous tissue is a neuron, and that a neuron is defined by a receiving end, dendrites, a sending end, the axon terminal, a synapse, that's the gap between the two cells, an axon that allows for the propagation of the signal, and an oligodendrocyte that provides the fat padding, which we'll learn later, allows for us to much more efficiently send signals down our neurons. So that's where we'll wrap it up. You're familiar now with the basics of nervous tissue, muscle tissue, connective tissue, epithelial tissue, and how they make up our different organisms and our different organ systems is what the vast majority of the remainder of the class will focus on. So please make sure that you reference the vocab sheet for this lecture and all lectures in the future, as it will pinpoint all the different particular organs and tissue types and cell types that I hope you retain from our lectures.